Hi, hello, and welcome. Today we will talk on the topic population genomics. Do anybody know what is population is? Population consists a group of the same species living in one area at the same time. Studying populations helps us understand how genetic traits are distributed and how they evolve over time. Next, we have demography. Demography is the statistical study of populations. It is used to predict how the size of a population will change. Let's see the three key features that population depend on mainly are population size, population density, and dispersion. First, we have the population size, where the total number of individuals in a population is much more smaller and are more prone to extinction due to random events like natural disasters, inbreeding, which can increase the appearance of recessive traits, reduce genetic variability, which limits the adaptation to changes, and also, population size is influenced by density dependent factors such as disease or food availability, and also density independent factors such as floods or climate changes. Next key feature is population density, where the number of individuals in a specific area has low counts. Due to this, the individuals have fewer encounters between one another and leads to lower reproduction rates. Lastly, we have dispersion, where the arrangement of individuals within a population are distributed randomly, uniform, or clumped. Let's see how population growth is predicted using two primary models. Key factors that influence population growth are birth rate, death rate, and carrying capacity. First model is exponential growth. It occurs when resources are abundant. The growth rate is calculated as R equal to birth rate minus death rate, where R is the rate of growth. Second, we have logistic growth. This model considers resource limitation. Growth stops when the population reaches its carrying capacity K, which is the maximum population size the environment can support.
Next is role of natural selection in biodiversity. Natural selection favors traits which is alleles that enhance survival and reproduction. The importance of allele frequency and genetic diversity in evolution. The first one is allele frequency. The frequency with which alleles of a particular gene are present in a population. Next is Hardy-Weinberg law, which is to estimate allele frequency within populations. And lastly is genetic diversity. Population with more genetic variation adapt better to environmental changes.
The Hardy-Weinberg principle states that the genetic makeup of a population remains constant across generations unless evolutionary forces act. For a population to remain in Hardy-Weinberg equilibrium, it must meet five key assumptions. The first one is the population is large to prevent random genetic drift. The second one is random mating occurs, meaning no mating preferences exist. No mutation occurs, so genetic information remains stable. No migration, ensuring no gene flow into or out of the population. No natural selection, meaning all individuals have an equal chance of survival and reproduction. Under these assumptions, the principle allows us to predict alleles and genotype frequencies using mathematical formulas. The allele frequency equation P plus Q equals to 1, where P is the frequency of the dominant allele and Q is the frequency of the recessive allele. The genotype frequency equation P square plus 2PQ plus Q square equals to 1, where P square is the frequency of homozygous dominant individuals, 2PQ is the frequency of heterozygous individuals, and Q square is the frequency of homozygous recessive individuals. Now, let me look into the impact of natural selection and genetic drift. First of all, what is natural selection and fitness? Natural selection is a selection by environment. where fitness is a measure of the relative survival and reproductive success of a specific individual or genotype. Selection against dominant allele can be complete in one generation, while selection against recessive allele need to be complete in many generations. It is because the recessive alleles are not expressed in heterozygous. They were protected from selection. Selection has a little effect against recessive allele frequency, while it has a greater effect on dominant allele frequency. And it also gives an intermediate effect to the alleles with incomplete dominant or adaptive effect. Therefore, deleterious recessive alleles can persist for many generations and useful in evolution. At the same time, it is beneficial in environment or in combination with other genes. One classic example is sickle cell anemia caused by the HBS allele. The dominant allele HBA produces normal hemoglobin, while the recessive allele HBS causes sickle cell anemia. However, individuals with the heterozygous phenotype HBA-HBS are resistant to malaria. This provides a survival advantage in regions where malaria is common, allowing the HBS allele to persist in the population. In conclusion, selection affects the variation in population, allele frequency of genetic disorder, and fitness of an organism. Finally, let's discuss the genetic drift and the gene pool. What is gene pool? Gene pool is a set of genetic information carried by the members of a sexually reproducing population, while genetic drift is a change in allele frequency by chance and not by selection. Genetic drift has a big effect on small, isolated population. This effect can be grouped into two types. The first one is temporary changes, where allele frequency fluctuates over time, and the second one is permanent changes, such as fixation of an allele, where allele reach a frequency of 1.0 while others are eliminated.
The application of SNPs are vast and transformative. In disease association studies, SNPs help identify genetic risk factors for conditions like cancer, diabetes, and heart disease. They also play a key role in personalized medicine, enabling tailored treatments based on an individual's genetic profile. Beyond medicine, SNPs are vital in trait mapping, linking genetic variations to physical or behavioral characteristics. They also shed light on evolutionary history, tracing human migration patterns and genetic diversity across population. Okay, so for the last point is exploration of GWAS. Genome-wide association studies, or we call as GWAS, leverage SNPs to identify genetic variants associated with specific traits or disease. So before we start, we should know what is SNPs. SNPs is the DNA that the blueprint of life hosts countless variations that make us unique. Among these variations are single nucleotide polymorphism, or SNPs. SNPs are the most common type of genetic variation occurring when a single base in the DNA sequence is altered. They serve as powerful genetic markers, enabling researchers to uncover the genetic basis of disease, traits, and population diversity. Evolutionary history, tracing human migration patterns and genetic diversity across population. These applications demonstrates how SNPs are unlocking the secrets of our genetic code, driving innovation in health, science, and beyond. So for the single nucleotide polymorphism, or we call as SNPs, are not just tiny changes in DNA, but they are keys to understanding life itself. By combining advanced technologies with innovative study designs, SNPs continue to propel discoveries in genetics and shape the future of science.
Now let us focus on component of GWAS. This is a flowchart of the component. It starts with input data, where we need individual and genetic markers. Then we proceed with association method, where we need to adjust for confounders and add on the level of complexity. Next is validation replication, where we can identify same and dissimilar conditions. And last process is interpretation, where we can see the functional analysis and follow-up studies. Got three components of GWAS study design: study scale, marker design, and subject design. For study scale of genetic, we focus on candidate genes, which we call as hypothesis driven. For genomic is hypothesis free. For marker design, we need to select informative markers, which is common variance SNPs versus rare variance MEF below 1%. And then we need to choose the platform of the accuracy, error rate, and marker distribution across the genome. And lastly is a subject design.